Welcome to G Fun Facts Online. Today, we are dispensing with the pleasantries to drop you directly into a nightmare scenario. Imagine a world where the water is as thick as syrup. The sun beats down with an intensity that scours the landscape, and the chemistry of the environment is so hostile that it would strip the water right out of your cells, leaving you as a desiccated husk in a matter of seconds. This sounds like the surface of a distant, uninhabitable exoplanet, but it is not. These are the hyper-saline environments of Earth. The salt flats, the deep-sea brine pools, and the neon-pink lakes that defy our intuitive understanding of what constitutes a habitable zone. For centuries, humanity viewed these landscapes as barren wastelands. We named the Dead Sea for a reason. We looked at the crystallizer ponds of industrial saltworks and saw nothing but chemical basins. Yet, under the microscope, these environments are teeming with life. They are not just surviving. They are thriving in conditions that push the absolute limits of biochemistry. This is the world of halophiles, the salt lovers. Their existence is one of the most remarkable tales of adaptation, evolution, and biological ingenuity on our planet. Over the next 20 minutes, we are going to tear apart the molecular machinery that allows life to function in saturated salt, tour the most exotic locations on Earth, and reveal how these extremophiles are reshaping biotechnology and our search for extraterrestrial life. To understand the biology of halophiles, we must first understand the enemy they face. That enemy is salt. Sodium chloride is not merely a seasoning. In high concentrations, it is a potent destroyer of biological structure. The primary challenge in a hypersaline environment is osmotic pressure. Water naturally flows from areas of low solute concentration to areas of high solute concentration to equalize the balance. For a typical cell placed in a brine pool, the environment is hypertonic. The physical laws of the universe demand that the water inside the cell, the solvent of life, rush out into the brine. Without specific adaptations, a cell in this environment undergoes plasmolysis. Its membrane pulls away from the cell wall, its cytoplasm shrinks, and its metabolic machinery grinds to a halt. The DNA precipitates and the cell dies. But the challenge goes beyond simple water loss. High concentrations of ions are toxic. They interfere with the hydration shells of proteins, stripping away the water molecules that keep proteins dissolved and functional. This phenomenon, known as the salting out effect, causes proteins to clump together into useless aggregates. To survive here, an organism must not only hold onto its water with an iron grip, but also redesign its entire molecular architecture to function in a soup of ions. Biologists measure the availability of water using a parameter called water activity. Pure distilled water has a water activity of 1.0. Seawater is approximately 0.98. Most common bacteria, like E. coli, cannot grow below 0.91. Hypersaline environments push this number perilously low. A saturated sodium chloride solution has a water activity of about 0.75. Some magnesium-rich brines go even lower. Halophiles have conquered these low water activities, with some specialized fungi and archaea capable of metabolism at values near 0.61, a limit often considered the absolute floor for life as we know it. Evolution has devised two distinct, divergent strategies to solve the salt problem. These strategies represent a fundamental split in the tree of life, dictating everything from the organism's bioenergetics to the very sequence of its proteins. The first is the salt-in strategy. This is the more radical of the two solutions, primarily employed by the archaea, specifically the Haloarchaea, and a remarkably convergent bacterium called Salinobacter. Instead of fighting the salt, these organisms embrace it. They do not pump salt out, they let it in. To balance the massive osmotic pressure of the outside world, they accumulate potassium chloride in their cytoplasm to concentrations that match or exceed the external salinity, often reaching 4 to 5 molar. This creates a new problem. The intracellular machinery must now function in a solution that is essentially concentrated potash. A normal protein placed in 4 molar potassium chloride would instantly denature. Consequently, the salt-in strategists have undergone a massive, genome-wide evolutionary overhaul. If you analyze the proteins of a salt-in halophile, you will find a striking anomaly. They are overwhelmingly acidic. The surface of these proteins is covered in negatively charged amino acids, like aspartic acid and glutamic acid. In a high salt environment, these negative charges interact with the cloud of positive potassium ions and water molecules, creating a hydration shell that keeps the protein soluble and flexible. However, this adaptation comes at a steep price. These organisms are now addicted to salt. If you place a Halobacterium cell in fresh water, its proteins, adapted to be stable only in high salt, will mutually repel each other due to their negative charges. The cell wall will disintegrate, and the cell will burst. They are prisoners of their own adaptation. The second approach is the Compatible Solute strategy. Most halophilic bacteria, along with algae and fungi, take this more flexible approach. They maintain a cytoplasm that is relatively free of salt. To balance the external osmotic pressure, they synthesize or accumulate massive amounts of organic molecules known as compatible solutes, or osmolytes. These molecules are called compatible because they can exist at extremely high concentrations without interfering with the cell's delicate enzymatic machinery. Common compatible solutes include amino acid derivatives like proline and glycine betaine, sugars like trehalose, and polyols like glycerol. These molecules work by preferential exclusion. They are excluded from the immediate hydration shell of proteins, which forces the water structure around the protein to remain intact, stabilizing the native state of the protein. Unlike the salt-in specialists, these organisms can often survive in a wider range of salinities. If the external salt concentration drops, they can simply break down or excrete the solutes. It is an energetically expensive strategy, as synthesizing these complex molecules costs a lot of ATP, but it provides ecological versatility. Now that we understand the mechanics, let's meet the cast of characters. The world of halophiles is populated by a bizarre cast of microscopic entities. Perhaps the most famous is Halobacterium salinarum. Despite its name, it is not a bacterium at all, but an archaeon. It is the organism responsible for the purple or red hue of salted fish and solar salterns. It possesses one of nature's most elegant inventions, bacteriorhodopsin. This protein acts as a light-driven proton pump. When oxygen levels in the brine drop, which happens often as warm, salty water holds very little oxygen, this organism switches to a primitive form of photosynthesis. It uses light energy to pump protons out of the cell, creating a gradient that drives ATP synthesis. It captures sunlight, but without chlorophyll. To ensure it stays in the photic zone where the sunlight is, it produces gas vesicles, proteinaceous, gas-filled balloons that grant it buoyancy. Then there is Haloquadratum walsbyi, the living tile. For decades, microbiologists were baffled by square bacteria seen in microscopy samples. In 2004, the organism was finally cultivated. Its cells are perfectly flat, square tiles. This extreme surface-to-volume ratio is an adaptation to nutrient limitation in dense brines. These cells often link together to form large, fragile sheets that float on the surface of magnesium-rich brines, looking like sheets of microscopic postage stamps. To prevent complete desiccation, they secrete a giant protein called halomucin, which holds a layer of water around the cell like a microscopic wetsuit. We also find the imposter, Salinibacter ruber. This is a bacterium that thinks it is an archaeon. Living in the same crystallizer ponds as Halobacterium, it has evolved to look and act almost exactly like its archaeal neighbors, using the salt-in strategy and accumulating potassium chloride. It is a stunning example of convergent evolution. And we cannot forget Dunaliella salina, the beta-carotene factory. This unicellular green alga is the reason many salt lakes look pink or orange. When exposed to high light and high salt, it pumps its chloroplasts full of beta-carotene, the same pigment found in carrots, to act as a sunscreen against UV radiation. To truly appreciate these organisms, we must visit their homes. Let's start with the Dead Sea. It is a terminal lake, meaning water flows in, but leaves only by evaporation. For years, the Dead Sea is relatively clear, but after rare, massive winter rains, the top layer gets diluted, triggering a biological explosion. Dunaliella algae bloom first, turning the water green. Weeks later, the lake turns a deep blood red as the archaea bloom in the trillions, feasting on the algal leftovers. The Dead Sea is unique because it is rich in magnesium and calcium, not just sodium. Magnesium is chaotropic. It destabilizes biological molecules even more than sodium, limiting life to only the hardiest specialists. Next, we travel to the Danakil Depression in Ethiopia. This is a poly-extreme environment, hot, acidic, and hypersaline. Researchers studying the hydrothermal pools here have found places where no life exists. The combination of high temperature, acidity, and chaotropic magnesium salts creates a barrier that even extremophiles cannot cross. This zone of sterility helps astrobiologists define the theoretical limits of life. Yet, just adjacent to these sterile pools, unique, nano-sized archaea cling to existence. Then there are the deep sea brine pools. Deep in the ocean, distinct pools of water sit on the seafloor, refusing to mix with the seawater above. These are deep, hyper-saline anoxic basins. The boundary between the seawater and the brine is a sharp halocline, a trap for organic matter falling from above. The brine is so dense, oxygen cannot penetrate it. The organisms here are anaerobes, evolving in complete darkness and isolation for thousands of years. Wherever there is life, there are viruses, and hypersaline environments are dominated by them. Haloviruses often have morphologies seen nowhere else, such as the lemon or spindle shape. This shape is thought to be an adaptation to the high viscosity of the brine. Some, like the Acidianus two-tailed virus, can change shape outside the host, growing two long tails to act as grappling hooks. In a saltern crystallizer pond, viruses are the engines of the ecosystem. They facilitate nutrient turnover by lysing cells, releasing carbon and nitrogen back into the soup to feed the survivors. The unique adaptations of halophiles have turned them into gold mines for biotechnology. If a protein can remain stable in saturated salt and high heat, it is virtually indestructible in an industrial setting. Take ectwine, the compatible solute produced by Halomonas elongata. It binds water molecules so effectively that it is used in cosmetics as an anti-aging ingredient and in medicine to stabilize cell membranes. Industries use a process called bacterial milking to harvest it. They grow the bacteria in high salt, then shock them with fresh water. The bacteria, sensing the osmotic drop, dump their ectwine to avoid bursting. They are then returned to salt to start the cycle again. Then there is Bacteriorhodopsin, the purple protein. It is photochromic, changing color and shape when hit by light with incredible speed and durability. Scientists have used it to create holographic memory cubes and are testing it for artificial retinas. And we must mention bioremediation. Halophiles are being deployed to clean up oil spills in marine environments, as some species feed on hydrocarbons, even in high salinity wastewater, where normal bacteria would die. Finally, the study of halophiles connects us to the cosmos. We know Mars was once wet and salty. As it dried up, it likely formed Hyper-Saline lakes. If life existed there, it would likely have been halophilic. Furthermore, halophiles can survive trapped inside fluid inclusions in salt crystals for millions of years. Finding salt crystals on Mars could mean finding time capsules of ancient Martian biology. We also look to the moons of Jupiter and Saturn, Europa and Enceladus, which hide vast liquid water oceans beneath miles of ice. These oceans are briny. The strategies we see on Earth are the most likely blueprints for life in these alien seas. However, this resilience poses a risk. Because halophiles are so resistant to radiation and desiccation, they are the organisms we most fear accidentally sending to Mars on a rover. If a Halobacterium hitched a ride, it could potentially survive the journey and contaminate the Martian brine. Extremophiles challenge our anthropocentric view of nature. We tend to think of extreme environments as marginal, difficult places where life struggles to exist. But for a Halobacterium in a sun-drenched salt pond, or a Haloquadratum floating in a magnesium brine, this is paradise. They teach us that life is not fragile. It is robust, adaptable, and incredibly creative. By understanding the biology of the Hyper-Saline, we unlock new technologies for our future, gain a window into the ancient history of our own planet, and prepare ourselves to recognize the face of life when we finally find it among the stars. The salt of the earth is not barren. It is one of the most vibrant stages for the dance of life. Thank you for listening to G Fun Facts Online.